Okay, so today we are going to begin the topic of monoclonal antibodies. Basically, the concept of monoclonal antibodies comes from the problem of getting the anti-serum. Yesterday, I told you that what is an anti-serum? The anti-serum was the solution that was containing the antibiotic. So
Yes, yes.
initially it was like it was seen that many times in the very beginning it was very difficult to get the antibodies because to get the antibodies you have to make someone sick. You have to make someone sick, otherwise the antibodies, obviously when the primary immune response was would not be shown, then obviously it is a very obvious thing that there would be no antibodies. So that is why a person has to get sick
Mm.
to extract the antibodies out of that person. Now what happens that many scientists came up with an idea, and they thought that maybe if we use the antibodies, if we use the animals to you can say
Extract the
make,
antibodies from.
yeah, to make the antibodies for us, and obviously we would later we would extract it. But the problem with it was that animal rights issue obviously were there, and initially when we started these experiments, we injected the vaccines in the horses, monkeys, and in the rabbits, etc. But there was a problem with the animal rights, so we have to abandon those practices. The real thing is like obviously it's also an issue of animal rights, but with the mice, you can say. It is very ironic to say, but it is a reality that for with respect to mice, the laws are not that much strict compared to those animals which are more
Mm.
frequently pet by different people, like the
Livestock as
monkeys, the
well.
horses. So that is why we started using the mice. And what we actually do, we take the antigens in a in a very small amount. We take the antigen. Now this antigen basically is a virulent pathogen. Now what is the meaning of a virulent pathogen? This word virulent does not mean virus. The virulent basically refers to a pathogen that can cause disease. It
Okay.
can cause disease. So we like it is active, because the principle in according to the principle of vaccine, when we inject the antigens, we always use the weakened pathogen or dead pathogens. But a virulent pathogen mean that we inject the pathogens which are completely active, able to cause the disease. So we inject those pathogens into the mice. The mice respond, and they start producing the plasma cells. And these plasma cells are mostly stored in the spleen of mice. Why spleen? Remember that spleen is supposed to be a lymph node. Now, what is the meaning of lymph node? Lymph node means that every lymph node of our body, there are thousands of lymph nodes in our whole body. So,
Got it.
every lymph node in our body contains the lymphocytes, and the spleen is one of the largest lymph nodes. So
Okay.
the scientist injected first of all, the whole process is although not mentioned in your book, but the antigens or pathogens were directly injected into the spleen of an animal. So you can
Okay.
simply say that highest number of plasma cells would automatically expected to be found in the spleen. So spleen is creating the plasma cells, and then initially the plasma cell would produce antibodies, and we then use to extract them. We
Okay.
use to extract them. The problem is that This particular phenomena, in this particular phenomena, we have certain problems. What is the problem? The problem is that animals' lifetime of animal. Then there may be some issue with like the antibody rejection. Like
Yes.
obviously I already have told you yesterday
Yes, sir.
that you are going to get an immune response, um, uh, uh, you can say as a reaction against the sugar molecule positioning of on the antibody molecule. So simply putting
Yes,
it,
sir.
that if you would take the antibody directly out of the mice, it means that you are going to get the antibody that is not human made. And that would be the problem for our body, and our body would start producing it would start reacting in the
Exactly,
form of
yes.
an immune response. So it would react. So that is why the antibody would be rejected, and ultimately, the problem could get more even severer compared to the previous one. So we have to come up with a plan. We have to come up with a plan to avoid these two big problems. Number one, lifetime of animal antibody rejection and the animal rights as well. Like minimize the use of mice also. So the concept about the monoclonal antibodies finishes here. Now a new concept is about to start. But let me clear one thing. I need to clear this thing has to be cleared very, you can say, in a very straightforward way. What is the actual meaning of monoclonal antibodies? We know what
Yes,
is
sir.
monoclonal. Remember that the monoclonal is a compound word. Mono means single, clonal mean clone, meaning antibody related to a single particular clone of lymphocytes. We know there are many clones, millions of them. We have millions of clones of B
Mm.
lymphocytes and
So
T lymphocytes.
it has a specific antigen on it. This lymphocyte has a specific antigen on it, right?
No, 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 no. These lymphocytes are supposed to be, these lymphocytes or plasma cells are supposed to be related to a particular B lymphocyte that would be creating particular antibodies. Remember, it is not antigens. The
Yes, sir.
antibodies, these are not antigens. These antibodies are going to relate it to that particular clone which is working against a particular disease.
Okay,
So
sir.
suppose that, like yesterday, you you were injected or you were exposed to a pathogen, and you showed the
Okay.
primary immune response. Your B lymphocytes started producing the antibodies. Now we know that once a B lymphocyte is selected, once the clonal selection occurs, then the next step is clonal expansion, right? So when the clonal expansion occurs, it means
Yes,
that
sir.
only that particular B lymphocyte would be expanded or would be increased in number, to which the antigen has been exposed. The others would
Okay.
not be multiplied, they would not be expanded. Remember?
Yes, sir. Yes.
Now, the same thing
It's
we are
clear, sir.
doing here. I'm sorry, you said something?
It's, I said that it's clear now.
Okay, so the concept of monoclonal antibodies is that this antibody would be related to a single particular clone of B lymphocyte.
Okay, sir.
Like there are millions of clones, but we are making the antibodies from only one
That
clone.
particular
So
one.
you can,
Okay.
yes, the origin is specific. Origin is specific. It's not random antibody. We know that it is made from the particular origin. That is why we say monoclonal. It is related to that single clone. This antibody is related to that single clone made in the body of mice. So this was the concept of monoclonal antibodies. Just summarizing it, it was a problem in making the anti-sera. So we started injecting the animals because it was very, very dangerous sometimes. Like if we inject a human directly with the rabies virus,
Yes, sir.
back in those days, obviously it is a thing of, let's say, 50 years back. So back in those days it was like not the animal rights were not that much hard, but injecting a human being for the purpose of using him or her as a tool of science was
Producing
strictly
antibody.
prohibited. Yes,
Yes, sir.
so
Okay.
it would be very much riskier. So that is why we started putting the antigens into the animals, and then later we started getting the antibodies. And we, when we extract it, we would be able to inject it and save the life of the human being. And then I told you the source of this word and the reason of this word, monoclonal antibodies. Okay, so now
Yes.
the basic thing about the
Okay.
antibodies that they can be prepared from the animals, but the problem is the animal rights are against it, the lifetime of animal, the antibody rejection by the human being. These all things call for something through which we can avoid these issues, we can avoid these complaints, these objections. So we suddenly came up with a plan, and that was like, I have to say that it was a plan of a genius. And like they came up with a plan of a
Hmm.
hybridoma.
Okay. Okay.
Hybridoma. A
So,
hybrid.
uh,
Hmm.
so in the previous slide, you said that the lifetime of animal. Can you
Hmm.
explain this point further? What does it
Like,
mean?
suppose that you have injected a mice with an antigen, and the mice is producing the antibodies, and you are extracting the antibodies out of the mice's spleen. But
Yes, sir.
you know that after some time, and especially when the body is doing such a hard work, the lifetime of the mice is, or the animal is, at stake.
Okay.
Like,
Okay. Okay.
it's like you can obviously say that if a person is continuously sick, then the person's lifetime, the person's behavior, everything is at stake. So this is the same thing, that the animal is going to die very soon, and we have to inject new animals or new mice with the antibodies. Obviously, scientists don't work on a single mice. They often work
Yes.
on 1,000 mice samples. But the problem is that the supply of antibodies and this kind of thing is so much in demand. The immediate antibodies are so much in demand that you cannot fulfill this throughout the world. That is why that is why you have to come up with a plan in which you use the mice, you start using the mice minimally, and
Okay, sir.
you can overcome these shortcomings.
Okay.
So, for that purpose, the scientists came up with the idea of hybridoma. It is a hybrid of or between a plasma cell and a cancer cell. This is the
Okay.
definition. But now let's see what are the problems here. See, let me give you some information. The plasma cell have a problem. What is this? They have a problem of being short-lived. They are short-lived. They are going to be, the plasma cells can never survive for longer. Suppose that
Yes, sir.
if you tackle the disease, the plasma cell may stay in your body for 4 to 6 months. It is the memory cells
Hmm.
that can stay for 20 years or above.
Okay, sir.
So, and the second thing about the plasma cells is that they cannot divide. They cannot
Okay.
divide.
We cannot do it.
And they cannot produce antibodies in vitro. What is the meaning of in vitro? In vitro, the word in vitro is a very specific word that we refer
Okay.
to when we say something occurring outside the cell or outside the body.
Okay.
That is
Okay, sir.
in vitro, or in the lab, in the laboratory when we do some experiment um, outside the body, we say that it's in vitro, or maybe outside the cell. So the plasma cells cannot produce antibodies in vitro.
Okay, sir.
Now, about the cancer cell, obviously the cancer cell, notorious ones. The problem is many type of cancer cells have an unlimited telomerase activity,
Yes, sir. Yeah.
and for that reason you cannot finish them, you cannot avoid them. And there is a type of cells you can search for it on the Google. There was a female whose names were starting the starting of his name, the first name was AHE, and the second name, or maybe the last name, was starting from
Okay.
LA. So the
Okay.
scientists used these four alphabets, and they named her cancer cells as HeLa cells. These cells are
Okay.
still dividing since 1952.
Okay. Do we
They
have
are,
those like in the exhibitions or the museum?
yeah, we have those in the scientific museums, but not here in Pakistan. But
Okay, sir.
maybe the British, and they also filed a claim, by the way, later, I guess after 2000, that it was totally without the consent of the female and the family of that female. So, but obviously they have to give them fine or whatever the agreement was there.
Mm.
But these cells are still there and still dividing.
Okay.
Now, the cancer cells, some of the cancer cells have this much higher telomerase activity. And
Yes,
you
sir.
should also know that some cancer cells uh, perform function, perform their functions. Like, let me, while
Actually, class with.
being cancerous,
So
while
can
being
you repeat that?
cancerous. Yeah, sure, I can. Some cancer cells perform their functions while being cancerous, and they sometime overperform. No, not sometime. They overperform the function, overperforming the function. Now, what does it mean? Suppose
Sir,
that,
metabolic
have
rate
you,
how?
yes, metabolic rate. There is a problem with the thyroid. The thyroid cancer occurs, and when the thyroid cancer occurs, the cells that produce thyroid, they get cancerous, and they start producing so much thyroid that that thyroid level gets out of the range of homeostasis of the body. So that person, due to that higher amount of thyroxine being produced by thyroid, the people get heart attacks. They start becoming very weak. Why? Because the thyroxine would cause a higher rate of respiration in your body, causing the heart to
Mm.
beat very fast, ultimately leading it to the attack. And the reserves of the body would be utilized. So some cancerous cells don't perform any function, and some
Mm.
perform the function at such a higher rate that it gets out of the control of the body. So the same thing happens with the cancer cells. Now, coming back to the hybridoma, the problem with the plasma cells and the qualities of the cancer cells, what did the scientists do? Through some technique, they were able to uh, fuse a cancerous cell and a plasma
Plasma
cell.
cell.
When
Sir,
they
do we have to know the
produce,
diagram?
um, do we? Sorry,
Do we have to know the diagram of this and the specific
no.
diagram?
You don't have to, but some things, let's suppose they may give you this diagram, and they would be presenting, they would be labeling this with an X. Now you
Hmm.
have to be familiar that what would be here. This is like obviously common knowledge of
Yes.
hybridoma. So now when you would fuse a plasma cell and a cancer cell, it would become a hybridoma. Okay, now remember that most of the cancers have a word oma in at the end of them. At the end of the word, like at the end of the cancerous disease. Like there is
Okay.
a benign cancer of fat tissue called as the lipoma. And there
Okay.
is a very dangerous type of cancer of lymphocytes called as lymphoma. So most
Okay.
of the time when you refer to word oma, it refers to the cancer, but it does not mean it would always be metastatic or benign. Sorry, or malignant. But yes, it could be a tumor. So tumor is always referred by the suffix oma. Anyways, so hybridoma is made. Now, what shortcomings you can overcome here? Number one, you can perpetually produce antibodies in the labs.
Hmm. Um, but you said with the plasma cell, you cannot, it cannot produce the antibodies outside. But when it is in the hyperdoma condition, it can.
It can, yes.
Okay.
Now, what is the meaning of why it became perpetual? Due to high telomerase activity.
Okay, sir.
And sorry, high telomerase activity. And the word produce refers to it is also because of cancer cell. Cancer cell. So now you can overcome the shortcoming of the two shortcomings of plasma cell. Number one, they were short-lived. Number two, they could not divide. And
Okay.
number three, three shortcomings. All three shortcomings, by just doing this thing, they can produce the antibodies in vitro outside the body.
Yes, sir.
So that is quite you can say killing many sparrows with one stone, if we talk in
Hmm. Yes.
the language of idioms. So number two, like obviously we can overcome the complaint of um, animal rights,
Yes, sir. Okay.
because now we do not have to inject many mice with this particular antigen of animal rights. Why? Because once the hybridoma is made, you can at least produce the antibodies for many next years until the
Hmm.
conditions become unfavorable, or maybe the cell become exhausted due to some reason. You can overcome all those problems. Number three,
Okay.
obviously, uh, n- number of mice used for this purpose can be
Can be saved.
reduced. Yes.
So, is there any negative effect for this, like any adverse effect?
There was a negative effect, but obviously they did overcome that problem. What was it? When they fuse it, the hybridoma had the plasma cell from mice, and that plasma cell from mice was having a different sugar group
Hmm.
position on antibodies. The problem is this was causing the immune response. So what did they do? They genetically engineered they genetically engineered the hybridoma. Now this is the geniusness. They genetically engineered the hybridoma and install the gene or you can say added the gene for sugar molecule for sugar molecule location from human, taken from human.
Okay, okay, sir.
They took the sugar molecule location gene from the human and they
Okay,
added it in the plasma cell, leading to, or you can say, overcoming that shortcoming as well. So, overall,
okay,
the concept of hybridoma was
uh, sir,
revolutionary.
your voice is not clear. Sir, can you hear me?
Okay, sorry. Since when? It is
Hello?
till now. I can hear you.
Okay,
You
okay,
cannot?
okay. It cut in the middle, but it's okay. I understood the concept.
Okay, okay, okay. So basically, let me repeat my sentence. I was saying that this was a revolution in the field because we did not only use the monoclonal antibodies to prepare the anti-sera. We also use these antibodies, uh, like we also use these antibodies in diagnosis. We also
Okay,
used these in diagnosis. How? Now, there is a thing about the antibodies. Let me tell you about a concept of pregnancy strip,
sir, I
especially,
have a question.
or maybe yes, please.
Sir, is the hypo the one that we just did right now? Sir, can you hear
Hybridoma.
me? Hello?
Yeah, yeah, yeah. Yes, yes, yes.
Oh,
I can
sorry,
hear
it
you.
just keeps on cutting. Hybridoma, is it a monoclonal antibody?
Hybridoma is a cell. Hybridoma is a cell, and it is producing monoclonal antibodies.
Okay, sir. Yeah, okay, understood. Okay.
So basically, why it is monoclonal? Okay, now let me make it clear for you. Is my voice clear now?
Yes, sir, it's clear right now.
Okay. Hybridoma basically is a source of monoclonal antibodies because the plasma cell that you would fuse with the cancer cell, the plasma cell that you would fuse with the cancer cell is only one type of clone. It's only one
Okay,
type of clone. So that is why
okay,
it is a cell made to produce the antibodies from single origin.
okay, sir.
Okay, now the antibodies, we can use these antibodies for the diagnosis. Basically, we can make some antibodies fluorescent. We can make these antibodies fluorescent. What happens that these antibodies, if they would be joining with, you can say, with a complementary molecule, they would show a color change. They would show a color change. Now, what did we begin to do? We started making these antibodies immobilized on certain petri dishes, like the immobilized enzymes.
Yes,
We
sir.
started making them immobilized on certain dishes or maybe the test tubes. And when we started doing this, what can we do now? Now remember, these are these are non-fluorescent. Suppose that these are non-fluorescent uh, antibodies. Okay. You take now this thing that I'm telling you is not in your book. The procedure is not in your book.
Okay,
Just
sir.
to
But
make
it's in
the
the
things
syllabus.
clear. Yes. So just to make
Okay,
the
sir.
things clear, you take the patient's blood or the patient's tissue sample, and you put it in this plate. Okay,
Okay.
once you put it, you know that if the patient's sample would contain the complementary antigens, they would be joining with the antibodies.
Yes.
They would be
Yes.
joining with the antibodies. They would be joining with the antibodies. The whole solution
So I'm so sorry, I can't hear you at all.
to remove extra antigen.
Sir? Sir, may I rejoin? Hello?
Something wrong with the internet. I don't know why.
Hello, sir? Okay, okay, I will wait.
Hello.
Yes, sir,
I
now I
just
can
I
hear
just
you. I
changed
can
the
hear you
network.
right now.
I guess there is a problem on my side here. So maybe it's better now.
Yes, sir, it's clear,
Okay.
crystal clear right
So
now.
uh, the patient sample would be inoculated into the petri dish, and as a second step, we wash the whole solution. Why? To remove the extra antigens. We remove the extra antigens, and then now we would add in the solution like this is the solution, this is the petri dish, and these
Okay.
are the antibodies. Let me make it complete. The examples, basically it is some of the explanation is necessary to understand the examples that are given. So
Okay, sir.
now the antigens are here. Now you would add the fluorescent antibodies. New antibodies would be added now in the solution, which would be fluorescent. Now, if they attach to the antigens, now these antibodies are supposed to be complementary to the antigen that is added. Like these antibodies were basically taken from a particular source, and these are supposed to be complementary to the antigen from the patient sample. You
Okay,
getting my
sir.
point?
Yeah. Yes, sir,
So,
I got it point.
complementary to antigens. Okay, now these would be, if they attach, they would change the color of the solution.
So you're saying initially they were only non-fluorescent antibodies,
Hmm,
and then we added
hmm.
the patient sample, and then we're
Yes,
adding the fluorescent complementary ones.
yes.
Okay, sir.
The
Clear.
non-fluorescent ones were also complementary, but they were not
Yes, sir.
changing the color of the solution.
Okay.
So, but the fluorescent one, when they were added, now immediately after addition, the solution changed the color, and the color change represents a positive result for any disease. This kind of test is
Okay.
sometime it is also called as ELISA, and these kinds of tests are performed for cancers, for typhoids, for hepatitis, for HIV, even for HIV. You know that we now have a test, the strip test for HIV.
Okay.
You just give a person can just give the sample, and when the sample is inoculated on the strip, if the strip shows just like the pregnancy strip, if it shows the bands, then it means,
Mm.
God forbid, it would be the person would be suffering with the infection of HIV.
Okay.
Similarly, now it's not a matter of very, you can say, it's not a matter of a very long test. You don't have to wait for it. Similarly, pregnancy tests, the pregnancy strips are made on the same principle, in the very beginning. In the very beginning, the female's body, like after 2 weeks, maybe the female's body is releasing a particular chemical. That chemical could be captured by this method, and the pregnancy strip shows the positive pregnancy.
Sir, is it the
Many other,
hormone that are produced?
yeah, there is a hormone. The name of the hormone,
Sir,
it is HCG, human chorionic gonadotropin. Yes, you were saying something.
so I was asking, why are they non-fluorescent antibodies already present? Like, why is it necessary to uh, attach the ones, fuse the non-fluorescent ones and the human patient sample first?
See, see, to make the result very much confirmed, to make the result
Okay,
very much confirmed, what do? We first immobilize them, then we wash the solution.
okay,
We wash the solution to remove extra antigens that may be present in the solution. So the new antibodies should only be attaching to already attached antigen, not to any other particular antigen.
okay, sir.
To keep the things very clear, to keep the things in black and white, we do these things. And
Okay, sir.
one more thing, sometime, just like the enzymes can attach to other substrate, like the inhibitor, similarly, the antibodies mistakenly can attach to a wrong antigen. So for that purpose, we don't use only one Petri plate, or we don't use only one sample. We run
Okay,
this test at least in 2 to 3 samples, so that we are sure that the antigen that is causing the color change is the one that we wanted to find out, not any random one.
okay, sir. It's
So
clear.
to keep the things in black and white. Okay, moving to the book now, you can see that we can, yes, the antibodies can locate the deep vein thrombosis, and uh, mouse with human fibrin. Yes, now we can locate the deep vein thrombosis problem. This is basically a clot formation in the leg, a very dangerous situation, because that
Okay,
clot become embolus. Embolus means that it can move in your body,
okay.
and when it
Is it
moves,
thrombosis or
uh,
embosis?
the deep vein thrombosis.
Okay,
If it is embolism, then it means it would be, it is staying. I'm sorry, it is thrombosis. Yes, if it is thrombosis, it is staying, but it can turn into embolism.
okay, okay.
Embolism means that it would travel, and it can travel to the brain or even into a major artery or vein, and not letting the blood completely enter into the heart. A little
God
brother
forbid.
of my friend was, he died because of this thing, DVT.
That that's severe. Okay.
Yes.
I hope no one gets it.
So,
No one also.
yeah.
Okay.
Yes, as the maps carried out in the bloodstream, they bind. Yeah, this is the process that they are referring. What happens? The labeled antibodies are then introduced. These are radioactively labeled antibodies. Now we are moving, we are advancing with a very high pace. Like, don't only use the antibodies which are immobilized. Sometime we use the radioactive antibodies, and it is only possible if you get a lot of antibodies from a source, from a single source, and that is when you get the monoclonal antibodies. You get a lot of, now imagine if we were not able to get a lot of antibodies, from this source. Instead, we used to take a lot of antibodies from the mice. Imagine what would happen. We had to use the even we had to use the mice for the diagnosis purpose. That
Yes, sir.
is the thing that we have avoided now. So
Okay.
now the mice are basically okay. Sorry, what I was saying, that the antibodies are basically radioactively labeled. Then they are injected into the body. They reach to the site of they reach to the site of the fibrin, or you can say the clot. And you know that the clot is made of fibrin. Now, how would we know
Okay.
that the clot is there? Because the antibody would attach to the clot, and the concentration of antibody at that particular location would tell us that at this site the clot is really present. Because obviously,
Okay.
other than that, the antibody would not be present in your body in that high concentration, that much
Got
high
it.
concentration.
So where there's a wound, there is a lot of antibodies over there, no?
Mhm. They can be. No, no. Now, this is something this would confuse the things. If there is a wound and there is a clot, that is another case. That clot is supposed to
Okay.
be formed. In that
Okay,
case,
sir.
we would not go for this diagnosis.
Okay.
This is a diagnosis. This diagnosis is done when a person suspects that I am suffering with the DVT. So he would be injected
Okay.
with the radioactively labeled antibodies. The antibodies would reach at the site of the clot. They would bind to all the antigens present on the fibrins. When they would attach on the fibrins, and they can be seen in the radioactive sensors. It can be seen that antibodies are basically accumulated in the leg, because they would be
Okay.
releasing the radioactive very like you can say harmless, almost radioactive rays in the harmless
Okay.
concentration. You can receive them, and you can diagnose that yes, the clot is present, or no, the clot is not present.
Okay, sir.
So similarly, we are using monoclonal antibodies in treatments as well as in diagnosis. What treatments in which we are using it? uh, Yes, these 2 points basically are referring to the same thing I told you about the sugar chains and some amino acid problem, the sugar group and the immune response of humans. So we genetically engineered it to change the position of the sugar group, and we also changed a bit of amino acid sequence in the final antibody so that
Okay.
the human body does not react. Now,
Sir, so the difference is due to the sugar group in the antigen, right?
yeah, mostly in the antibody.
Okay. Okay, sir.
Hmm.
And in the antigens
Okay.
as well,
Mhm.
like the human antigens or only the antibodies?
Only see, we are injecting the antibodies from outside. So
Yes.
that particular molecule is specifically present on the antibodies. So we are only changing it on antibodies, not the human antigens.
Okay. Okay, sir. It's clear.
All right. So now let's read this. This particular part, this particular type of solution, this is used for the breast cancer genetic technology. It is mouse mab humanized mouse mab. What does it mean? We have genetically engineered it to make it look like a human antibody, but it is a monoclonal antibody that we took from mouse. So you can
Okay,
just
sir.
imagine why we are saying it humanized. Now this antibody binds to a receptor protein that is produced in abnormal quantities in the cell surface membranes of breast cancer, so we can easily identify it. Just imagine that we can, within the staying keeping the cells in the body of the human, we are able to identify any sort of cancer by using the
Mm,
radioactively labeled antibodies. So that is um, the
a
advantage.
milestone.
Hmm. The receptor protein is not unique to cancer cell, but cells within 10 to 100 times the usual number, these receptor molecule in cell surface membrane, can only be cancer cells. Now, this line, what does it tell? That receptor to which the antibodies The trastuzumab, basically the name of the antibody is trastuzu. The
Say,
mab
do
is
we have to
monoclonal.
individually revise these? Are these in the syllabus that we have to individual, like individually know what they do and how they perform?
Uh, Basically, they would be the names and the functions. They would be told, they would be mentioned in the exam. You just
Okay,
have to tell that how would they do that.
okay, sir.
Like they would reach to the side, attaching to the antigen, and then you can say we can detect them at radioactive detector and
Okay,
diagnose the disease.
okay, sir.
So these are basically just the examples. I was just going through them, so maybe you don't get intimidated by these examples or names.
Okay,
So basically, this particular antibody, which is attaching to the receptors, they are talking about those receptors, that those receptors are not specific to cancer cells. They can be found in your body, but above 100, 10 to 100 times the usual number of these receptor molecules can only be found
okay,
on the
sir.
cancer
Yeah.
cells.